Hi, my name is Anthony, and I'm a product manager at Millennium Science, and I manage Unchained Labs protein analytics instruments. So today I'm going to talk a little bit about one of those systems called the LUNATIC. So when I think about protein quantification, I think about it as a routine lab chore. You just have to get it done. It's like mowing the lawn. It's something that you need to do fairly often, and it's important to just go and actually get it done. However, using old school protein quantification methods is a bit like using a goat to mow the lawn. It works, but it's not exactly the fastest or most accurate way of getting your lab chores finished. The LUNATIC from Unchained Labs speeds up those lab chores with a plate-based high-throughput protein quantification system, and it uses extremely small sample volumes. LUNATIC requires only 2 microliters of sample, and it can measure up to 96 samples at a time in as little as 5 minutes using SBS formatted plates. So high LUNATIC plates, as shown here, are compatible with multi-channel pipetters and liquid handlers. They also include 2 fixed path length microcuvettes, which gives an extremely high accuracy and a broad dynamic range, all the way from 0.03 to 275 OD. So on this slide, when we zoom in on one of these wells in a LUNATIC plate, you can actually see what they look like. They look like a small microfluidic circuit. So LUNATIC is the only system out there that can measure proteins at both high throughput and high concentration. Each one of the wells is comprised of 3 parts: an input well, where you actually load the sample, a microfluidic reservoir, which holds the sample with no risk of cross-contamination or evaporation. And finally, you have the 2 fixed path length microcuvettes circled here, which is where all the measurements actually happen. So LUNATIC is very easy to use. You just drop and read. All you have to do is drop the sample into the input well, and then it's drawn into the microfluidic circuit storage channel by capillary action. So once the sample is actually in that storage channel, it can be left there for up to 2 hours without any detectable evaporation, which is actually pretty useful if, for example, you're waiting on protein samples to come off a column, or you've just started one part of the plate much earlier than you really needed to. So once you load this plate into the LUNATIC instrument, it will actually pump the sample from that storage channel into the microcuvettes, and that's where the UV vis absorbance reads occur. So here you can see the impressive range that's actually possible with the LUNATIC when using a real protein sample. This is an antibody dilution series in PBS with dilutions that range from absorbances up to 275 all the way down to 0.03 OD. So you might not be able to tell just by looking at these curves, but each one of these colored lines on the left-hand side is actually 8 replicate measures that are all overlapping. You can barely tell them apart because they're so close to one another, and this demonstrates how reproducible these UV vis spectrums actually are. If we take a look at just the 280 results, the A280 results on the right-hand side, you can see the high degree of reproducibility and linearity that LUNATIC gives you with protein samples based on the high R squared value, and the fact that although there are 8 points, at each one of those spots, you can't see them because they're overlapping so closely. So obviously these results look quite good, but you might be asking yourself, how good are they really? Can we put a number on LUNATIC's reproducibility? And the answer is yes, we can. LUNATIC has an accuracy of within 2% and a precision of within 1%. One of the ways that you can verify these values is with one of Unchained Labs standards, which is called the fundamentals. So the fundamentals is a set of 5 tryptophan standards at 5 different concentrations: 1, 2, 4, 6, and 8 milligrams per mil. These concentrations are independently certified, and each aliquot also has a color-coded label, which comes in handy for keeping everything straight. If you're more used to working with protein samples, you might be thinking that 1 milligram per mil seems a bit low. but that's because purified tryptophan has a much higher extinction coefficient, or E1%, than most other proteins. So the absorbance of a 1 milligram per mil tryptophan standard is about equivalent to a 20 milligram per mil IgG. And so the 8 milligram per mil standard has about the equivalent absorbance of a 160 milligram per mil IgG. So you can think of this as a way of covering the entire range of the lunatic from 20 to 225 OD. When you measure the fundamentals, you typically run 16 replicates to make sure that the lunatic system is performing as well as it can. So here you can see that setting up the fundamental standards is super easy, just like with all lunatic applications. Based on the color-coded labels, the lunatic software actually tells you where to load each standard, including the water blank within the 96-well plate. And it also gives you the input boxes on the right-hand side there, where you can enter the manufacturer's certified concentration for each ampule and the manufacturer uncertainty as well. So that's also slightly different for each ampule. So once the run is complete, checking the result is equally as simple because the lunatic does most of the work for you. So the lunatic software actually calculates the mean concentration, the mean recovery versus the certified concentration, and also the relative standard deviation, or RSD, of each standard. It also calculates the R squared value, or the linearity of all the samples taken together, and it tabulates everything for you here in this results table at the bottom of the slide. So tryptophan actually absorbs at the same wavelength maximum as proteins. And this set of standards is quite highly absorbing, which you can see on this graph. And that means that using the fundamentals, you can actually check that the system is performing in an absorbance and wavelength range that's most relevant to high concentration proteins without necessarily having to use one of your own precious high concentration protein samples. So you can just use the fundamentals instead of your own sample, which means that you're able to check the performance of the lunatic as often as necessary if desired. So all of this data that you see here from the software, you can actually export that into either Excel or a PDF, and then you can put it in your lab book if you so desire. So if we actually now plot the average results of each standard, as shown here on the left-hand side, you can see that that great linearity of the lunatic over the entire dynamic range. Both the trend line and R squared value are extremely close to 1, which again shows off that accuracy and linearity. And the spectra here on the right-hand side overlap perfectly for all 5 standards across that entire range. So not only do you get amazing linearity and reproducibility, but you also get precision and accuracy with the lunatic. Now, the average concentration, which is shown here as mean recovery, is all within 1% of the manufacturer concentration for each one of the standards. And more impressively here on the right-hand side where this arrow is, you can actually see that the RSDs are all less than 1%, even for the highest concentration sample. And this really shows off the lunatic's high degree of precision and reproducibility. So the nice thing about the fundamentals is that the concentrations and therefore the absorbances are all independently certified. So these standards can actually be used to compare the lunatic to other UV-Vis spectrometers. So here you can see a measurement for all 5 fundamental standards with 16 replicates for each one on the lunatic compared to a pedestal-based microvolume spectrometer. And you can see in every case that the green bars are a lot smaller than the blue bars. And that's because the lunatic measurements are closer to the manufacturer's certified concentration values for all 5 standards. The other system, represented in blue, sometimes underreported the concentration and sometimes overreported it. But the important thing is that the magnitude of that % difference was always larger with the other system compared to the lunatic. So standards are great, and tryptophan standards, as we've talked about, are quite useful since they absorb in the same range as proteins do. But the lunatic also has great performance with real proteins, which we'll go through now. So here we have a gravimetric dilution series of IgG in PBS, and it's measured with 8 replicates on the lunatic and on another microvolume spectrometer. So you can see that the lunatic trend line slope and R-squared value are both much closer to 1 compared to the other system, which indicates that the measured concentration is closer to the known concentration across the entire dilution range on the lunatic. If we look at the table on the right-hand side, you can see essentially the same thing broken down by sample. Lunatic was within 2% of the known concentration for all samples, and the other system was up to 6.1% difference in some instances. We also tend to see higher precision with the lunatic. So in this example, we have 4 different protein samples, each one at a slightly different concentration, measured on the lunatic and pedestal-based microvolume spectrometer. So the CVs, in this case, which is actually the same as RSD, were quite low for the lunatic, but the CV for the other system was anywhere from 4 to 16 times higher. So again, this shows that the lunatic is precise across both low and high concentrations across many protein types. So if speed is more important to you, lunatic still has you covered. Loading those 96 fundamental standard samples takes around 15 to 20 minutes using a single channel pipetta. It then takes another 9 to 10 minutes of automated read time on the actual system to get the results. But if you were to run these samples on another system, like a pedestal-based microvolume spectrometer, it can take over an hour because of all the hands-on time needing to clean the stage every time you pipette, et cetera, and so forth. So when you're comparing, and additionally, when you're comparing to other Fabret or cuvette-based systems, it takes around about a minute per sample, which of course for 96 samples will take up to over an hour and a half. So the lunatic gives you that speed that these other systems just don't. So for labs working at a much higher throughput and a much larger scale, the lunatic is also automation compatible because the SBS plates are compatible with liquid handling robots, and there are full robotic integration options with the software with an API. So the lunatic is downstream ready as well. And it uses something called the performance verification plate, which makes verifying lunatic performance for compliance with either the US or EU pharmacopoeia quite easy. Each one of these performance verification plates contains a set of independently certified NIST standards that are used for testing for compliance with all the requirements for UV-Vis spectrometers that are set out in those pharmacopoeia documents. So the lunatic sweet spot is, of course, if you're running lots of samples and need that reliability. And of course, accuracy and speed are important at every stage of the bioprocessing and anywhere really in the development process as well. So high speed, high accuracy protein quant is important anywhere from creation all the way up to quality control. However, if you're working more in the upstream or research-oriented setting that may have more complicated samples, lunatic has a few really cool tricks up its sleeve that are specifically designed for those circumstances. So one of those tricks, and my personal favorite, is the unmixed applications. Here I'll take you through a few examples and explain what they are and how they can be used. So a purified cell lysate or protein sample that's somewhere in the middle of the purification process can have a variety of impurities that will have an impact on the UV-Vis absorbance spectrum. You might measure a sample and get something that looks like this black plot here on the top of the screen. And maybe it has contaminants in it, maybe it doesn't, but the point is that you don't know that. Now, a traditional UV-Vis spectrometer would look at a spectrum like this and just interpret the entire A280 signal as being from protein, which may actually result in an over-reporting of the protein concentration. However, on the lunatic, the unmixed algorithms takes this black spectrum and actually breaks it up into its component parts. It actually deconvolutes it and reveals any contaminants. So the protein unmixed algorithm can actually identify proteins, nucleic acids, and other impurities, and it also deconvolutes the spectrum into a turbidity spectrum. Each of these deconvolutions are based on the characteristic spectrum of the chemical that it's looking for. So once the lunatic is done with these deconvolutions, using the unmixed algorithms, it then just quantifies the protein concentrations from the deconvoluted spectrum alone, which gives a much more accurate result. So here's an example of what this actually looks like in the real world for a protein sample analyzed with the unmixed application on the LUNATIC. The black line is the absorbance spectrum before deconvolution, and the unmixed algorithm identifies the contributions to the absorbance spectrum from protein, shown with the green line, and then from nucleic acids and other impurities, shown in the blue line. So you can see that the blue line itself actually has a peak around 260 nanometers, which is characteristic of nucleic acids. For most spectrometers, the A280 value for these impurities would actually get factored into the protein quantification. And so it would actually report the protein concentration as being much higher than they really are. So in this example, this sample contains a significant amount of nucleic acids. So depending on what it would be used for downstream, it may actually require some more processing or even some repurification. So LUNATIC works well for deconvoluting and separating out protein signal from nucleic acid signal, but it also sees through the turbidity of the sample. So when you look at a spectrum on the LUNATIC, it represents the turbidity as this gray background line. In this example, the turbidity is actually quite high. And high levels of turbidity in a protein sample or cell lysate can actually come from a few different sources. Lipid contamination and protein aggregation are pretty common, but there's also some potential for bead carryover or resin carryover from the purification process itself. Now, getting purity information at the same time as protein concentration is super useful, because not only does it help tell you if you actually need to do any further processing or repurification in order to get a protein sample cleaned up, but it can also tell you if it's even worthwhile to do it in the first place. For example, if a protein concentration is so low that it's not going to be useful anymore, then there's really no point in trying to clean it up and do all that extra work when you might actually be better off throwing it out and starting again. So here in this final example, we have a couple of examples that show how well this unmixed algorithm on the LUNATIC actually works with protein samples that have a few different spike-ins compared to a pedestal-based microvolume spectrometer. So on the left-hand side, we have 1 milligram per mil of HSA, our human serum albumin, with 25 nanograms of DNA spike-in. So you can see that the LUNATIC's unmixed algorithm has actually quantified it perfectly, and the other system actually over-reported the concentration. On the right-hand side, we have 1 milligram per mil of an IgG with 100 nanometer bead spike-in. And again, the LUNATIC's unmixed algorithm sees through the contaminant and turbidity and quantifies accurately compared to the other spectrometer system, which over-reported the concentration by a small amount. So even with dirty samples, the LUNATIC can still get the right answers. And the unmixed application is a tried and tested part of the LUNATIC that's actually been part of the software ever since the LUNATIC was launched back in 2017. So upgrading your protein quantification speed and accuracy with the LUNATIC from Unchained Labs makes your lab chores just that much better. And it's just like upgrading from that goat to a really nice lawnmower. Upstream or downstream, research or QC, the LUNATIC is just the best tool out there for doing your UV vis concentration. Thanks for your attention.